AppSec's online support program, Class 9, Week 10, Lesson 7, Subject Biology. Textbook Biology, page number 109 to 111, Unit 6, Enzymes, topic Uses of enzymes, factors affecting the rate of enzyme action. Objectives. By the end of the lesson, students will be able to revise uses of enzymes, that are food industry, brewing industry, paper industry and biological detergents, factors affecting the rate of enzyme action. Uses of enzymes. The first use of enzyme is in the food industry. Enzymes that break starches into simple sugars are used in production of white bread, buns, etc. It means that the starches or the carbohydrates, they are the complex form. So enzymes break these starches and the carbohydrates and they convert them into the simple sugars, and when they are converted into the simple sugar, so then they are used in the production of white bread, buns, etc. Brewing industry. Enzymes break starch and proteins. The products are used by yeast for fermentation. What does the term fermentation mean? Fermentation is actually an anaerobic process in which energy can be released from glucose even though oxygen is not available, and fermentation mostly occur in the yeast cells, right? So enzymes, I have already told you in the first point that enzymes actually break the starches and the proteins. When the starches and the proteins are broken down, so then the product that has been made, that product is used for the fermentation process or are used by the yeast for fermenting many different products, and the most common product of the fermentation is the alcohol. So mostly alcohol is produced in this process. Paper industry. Enzymes break starch to lower its viscosity that aids in making paper. Now, enzymes also help in breaking of starch, and when the starches are broken down, so they will lower the viscosity. What is meant by viscosity? Viscosity is anything that is thicker, right? Like you can see the honey, its viscosity is more than the water. So, when viscosity is lower, so then it will be helpful in making any of the paper. Biological detergents. Protease enzymes are used for the removal of protein stains from the clothes. Mostly we are having some stains on our clothes and that cannot be removed easily. For this purpose, we use the proteases enzymes only for the protein stains. Then amylases enzymes are used in dishwashing to remove resistant starch residues. Factors affecting the rate of enzyme action. Now, enzymes are very much affected by many of the factors like temperature, pH and the substrate concentration. And as you all know, enzymes are very much sensitive to the environment also. So, if any change occur in any of the factor, so it will affect the activity of the enzymes. So, we will be discussing some of the factors right now. Temperature. Temperature has the direct relation with the enzyme activity. If the temperature is increased, so it will also speed up the rate of the enzyme catalyzed reaction. But every enzyme works at its maximum rate at a specific temperature, right? Like enzyme has also some specific temperature. After that, the enzyme will not properly work out. And that specific temperature is called the optimum temperature. Every enzyme has its own optimum temperature, and within this optimum temperature, it speeds up its chemical reaction. So, when a temperature rises to a certain limit, heat adds in the activation energy, and you all know about the activation energy. I have already told you in the previous lecture that it is the minimum energy required to speed up any of the chemical reaction. So, heat also adds in the activation energy, and it also provides kinetic energy for the reaction. With the help of a graph, you can easily get this point that how with an increase in temperature, how the enzyme activity is increased. You can see that enzyme activity is increased up to 40 degrees centigrade. You can see over here, and this is an optimum temperature, right? After that, after 40 degrees centigrade, the enzyme will not properly do their work, right? So reactions are accelerated when temperature is increased, so reactions will be accelerated. When temperature is raised well above the optimum temperature, heat energy increases, the vibration of the atoms of enzyme and the globular structure of the enzyme is lost. Now, what happens if the temperature is raised from the optimum temperature? When the temperature is raised from the optimum temperature, so energy will be increased very much, and this increase in energy will cause the globular structure of the enzymes to denature, right? Denaturation of the enzyme will take place after 40 degrees centigrade or after the optimum temperature, and this is called the denaturation of the enzyme. The enzymes will lost its original shape, it will be D-shaped, and there may be sometimes the enzyme death is also caused when the temperature is raised from the optimum temperature. It results in the rapid decrease in rate of enzyme action, and it may block or it may be completely blocked. So I have told you that when the temperature is raised from its optimum temperature, so what will happen, that there will be a decrease in the rate of enzyme action. The enzyme will not properly work out, it cannot catalyze a reaction, or it may block that reaction. Now the second is the substrate concentration. Now how the substrate concentration affect the enzyme activity? If we are having enzymes molecule in any of the reaction and we are providing them the substrate, we are providing them more and more substrate, the substrate concentration is increased. So due to increase in the substrate concentration, the reaction will also be increased. But at a point when we are not having that much enzyme and the substrate concentration is increased, so there will be no more active sites to bound that substrate to that active site, so the reaction will not proceed further, right? If you are having in a reaction enzymes and you are having substrate, so you know that enzymes are having their active site and the substrate binds itself to that active site. But if you are continuously increasing the substrate concentration, so yes, it is obvious that the rate of reaction will be increased, but a point will be reached that there will be more substrate and there will be less enzymes, or there will be less active sites, so there will be no more active sites to which that extra substrate would bind itself. So at the end, what will happen, that the reaction will be stopped. So you can see in this slide that if enzyme molecules are available in a reaction, increases in substrate concentration increase the rate of the reaction, right? If enzyme concentration is kept constant and amount of substrate is increased, if you are keeping the enzyme concentration constant and you are constantly increasing the substrate concentration, so obviously a point will be reached that there will be no more active site to which that substrate will bind itself, right? All of the active sites will be occupied and any more substrate molecule do not find free active site. This state is called saturation of active sites and reaction rate does not increase. Okay, so actually when there are no more active sites available for the substrate, so that point is actually called the saturation of the active site, and at this point the reaction will be not proceed. Look at this graph. You can see in this graph that when the substrate concentration is more, so like we are having the active sites of the enzyme, we are having the substrates, so substrates are binding itself to the active site. In the second, you can see that all of the substrate has bind itself to the active site of the enzyme, and at the end you can see there are more substrate and all of the active site has been bounded by the available substrate and there are no more active sites for the other substrate to bind itself. So the reaction cannot be increased in this way. Come towards the pH. Now you all know that pH is the power of the hydrogen ion. You know it very well. So all the enzymes work at their maximum rate at a narrow range of the pH called the optimum pH. All of the enzymes are having their own pH. Some enzymes work at a higher pH level, some enzymes work at a lower pH level, right? We will be discussing some of the examples, right? So a slight change in this pH causes retardation in enzyme activity or blocks it completely. So if you are increasing or decreasing the pH level, so it can also retard the enzyme activity, or it can completely block that activity, right? So here are some examples. You can see that pepsin, which I have discussed already in the nutrition chapter in the digestive system, that pepsin is actually present in an intestine. Actually pepsin is present in a form of pepsinogen, but it is activated by the HCl. So this pepsin actually work in an acidic medium because its pH value is very much low. But if you look at to the trypsin, which is working in your small intestine, so it is working in an alkaline medium because its pH is very much high. So you can see that every enzyme has its own pH. It is working with its own medium. Like for the pepsin, it is working in an acidic medium, and the trypsin, it is working in an alkaline medium. Right? It is its nature. Change in the pH can affect the ionization of the amino acids at the active sites. If we are increasing or decreasing the pH value for these enzymes, so automatically it will have the direct effect on the reactions also. You can see in this graph, like this is the optimum pH for some of the enzymes. Like if you are increasing the pH, so it will be working more efficiently at 8 pH, but after that it will be not working properly. Now we will be discussing some of the characteristics of the enzyme. We have already discussed many of the characteristics of the enzyme in our previous lectures also. You can see the first one, almost all enzymes are proteins, that is they are made up of amino acids. I have told you that all enzymes are proteins, but not all proteins will be enzymes. So as enzymes are protein in nature, so they will be made up of the amino acids. You all know that amino acids will be forming long chains and they will be forming proteins, the complex structures. Okay? So most enzymes reaction rates are millions of times faster than those of comparable uncatalyzed reaction. This point has also been discussed before, that if any of the reaction is provided with any kind of the enzyme, so it will speed up the reaction, right? And as compared to those reactions which are not provided with any kind of the enzymes. Enzymes are usually very much specific for the type of the reaction and for the nature of their substrate. This point has also been explained. The enzymes are specific in nature because they are having specific active sites, so only specific substrate can bind, specific enzymes can come and bind to the active site of that enzymes. Only a small portion of enzyme molecule is directly involved in catalysis. This point have also been discussed earlier, that the enzyme are having their active sites and only the active sites are involved in the reaction. So this catalytic region is known as active site. Active site recognizes and binds substrates and then carried out the reaction. This point is also not new for you. I have told you that active sites actually bind the substrates to itself and then the reaction is carried out, then the products are formed, right? And there are also our enzymes activity is also regulated by some of the inhibitors and the activators. Inhibitors and the activators mean that there are some reactions, there are some enzymes which we want to inhibit them, or to which we want to slow down the reaction. We use inhibitors, and for some we need to fast them, so we will be using the activators. So the enzyme activity is actually regulated by these two, the inhibitors or the activators. Now another term came, that is the cofactors. Now there are some enzymes that will be working freely, like there is no need of any kind of component or any kind of protein to which enzyme attaches itself and it carried out its reaction. But there are some enzymes to which the additional component will be required, right? But if the additional component is not required to any of the enzyme to carry out its reaction, so that is called the cofactors. When any additional component is required to it, so these are the cofactors. You can see that some enzymes do not need any additional component to work, while others require non-protein part or ion. They are called the cofactors. Like Characteristics of enzymes. Characteristics of enzymes also include some of the organic cofactors and the inorganic cofactors. Organic cofactors actually included the flavins and the heme molecules, and the inorganic include the metallines. Now another word came, that is the prosthetic groups. When these organic cofactors like the flavin and the heme, when they are tightly bound to the cofactors, then they are known as the prosthetic groups. Right? And when they are loosely attached, when these organic cofactors like the flavin and the heme, when they are loosely attached to the enzyme, so then they are known as the coenzymes. So students, this was your today's lecture. Hope you find it easy and interesting. Thank you so much. Third phase, that is the anaphase. Actually, what happened in the anaphase? Here, the pulling of the chromosomes take place. You can see when a kinetochore spindle fibers connects with the kinetochore of the chromosomes, it starts pulling. As in the metaphase, you can see that the spindle fibers has attaches itself to the chromosomes at the point of centromere. Now when they attaches itself, so what will happen? They will start pulling in the opposite direction. The pulling force divides the chromosomes, sister chromatids, and they are pulled towards the respective centrosomes. Okay, so when the pulling is started, so the chromosome will be divided into the sister chromatids, and they will be pulled towards the respective centrosomes, as the centrosomes are present on the opposite poles. The other spindle fibers, the non-kinetochore, also elongate. Now what are the non-kinetochore? Non-kinetochore spindle fibers are those which do not take part in the attachment of the chromosomes, or the spindle fibers which are not attached to the chromosomes, they are actually called as non-kinetochore. And in anaphase, the non-kinetochore spindle fibers will be elongated. At the end, cells has succeeded in separating identical copies of chromosomes into two groups at the opposite pole. Now, as I have told you that the chromosome consists of two sister chromatids. When the pulling is started, so the chromatids will be detached. Half will move towards the opposite pole and half will move to the another opposite pole, and thus two identical copies have been produced.